Welcome back to the Deep Dive. One of the things I love most about doing this show is taking those complex scientific papers that you send our way and really unpacking them.
Yeah.
And today's episode is no different. It was actually inspired by some research that you, our listeners, shared with us about a pretty fascinating group of immune cells.
That's right, gamma delta T cells. They're a unique subset of lymphocytes, and they've been getting a lot of attention lately for their roles in both maintaining health and driving inflammation.
And you provided some really great material for this deep dive, some really in-depth scientific articles.
We'll be focusing especially on a recent publication, the role of gamma delta T cells in the immunopathogenesis of inflammatory diseases, from basic biology to therapeutic targeting.
That's a mouthful.
It is a mouthful, but it basically sums up what we're going to be talking about today.
Right. We want to understand how these cells, these gamma delta T cells, contribute to the body's normal functions and how they can become involved in diseases. Exactly. Like psoriasis and spondyloarthritis. Those are two big ones that we're going to be talking about today. And what's really interesting is that these diseases affect different parts of the body, right?
Yeah.
You've got the skin, the joints,
The gut.
the gut. So is there some kind of connection there? Could gamma delta T cells be the missing link?
That's a great question, and it's one that researchers are actively trying to answer. So to get started, let's talk about the basics of gamma delta T cells.
Okay, so where do these cells come from?
Well, like many immune cells, they originate in the thymus.
That's the little butterfly-shaped organ that sits right above your heart, right?
Exactly. And the research shows that gamma delta T cells are actually detected really early in development. In mice, they're seen as early as day 13 of embryonic development. And in humans, it's as early as the 8th week of gestation.
Wow, so they're there from very early on.
Yeah, and once they develop in the thymus, they don't just stay there, right? They migrate to different tissues throughout the body.
So they're kind of like little travelers.
They are. And what's interesting is that their migration is dependent on their T cell receptor or TCR chains.
So those chains kind of act like a GPS system telling them where to go.
Exactly. And based on these signals, they establish themselves as resident populations in various tissues, like the gut,
Okay.
the skin, the lungs, and even the reproductive epithelia.
So basically, places where our bodies come into contact with the outside world.
Precisely. Those barrier tissues.
So they're like the first line of defense.
They are. They're constantly on the lookout for danger, and they can react very quickly to danger signals.
So if they're so good at responding to danger, why do they sometimes cause problems?
Well, you see, that same responsiveness that makes them so good at protecting us can also backfire. Sometimes they can overreact and trigger an inflammatory response that actually ends up damaging the tissues they're supposed to be protecting.
So it's like they're a little bit too good at their job.
In a way, yes. And that's what makes them so fascinating to study, because if we can understand how they work, we might be able to harness their power for good and prevent them from causing harm.
Now, when we talk about gamma delta T cells, it gets a little bit confusing because there are different ways of classifying them in humans and mice, right?
That's right. In humans, we primarily classify them based on the V delta 1 or V delta 2 chains on their TCR, and there's a subset of V delta 2 cells that also have a V gamma 9 chain. So we call those V gamma 9 V delta 2 T cells.
Okay, that makes sense.
But in mice, it's a bit different. They use designations like V gamma 5 plus, V gamma 6 plus, and so on. And these different populations emerge at different developmental stages, and they populate specific tissues.
So it's not always a direct one-to-one comparison between human and mouse gamma delta T cells.
Right. But despite the differences in nomenclature, both human and mouse models have been invaluable for understanding the roles of these cells in health and disease.
Now, one of the things that makes gamma delta T cells really unique is that they don't always need to be activated by the traditional antigen-MHC interaction.
Right. They can also be activated by cytokines and other innate ligand receptor interactions.
So they have a more diverse set of triggers than alpha-beta T cells.
They do. And that makes them really versatile. They can respond to a wider range of threats.
The article mentions a couple of known gamma delta TCR ligands, SCINT1 and butyrophyllin-like molecules, or BTNLs.
Yes. And these ligands are thought to be involved in what's called normality sensing. Basically, by interacting with these molecules, gamma delta T cells are constantly being informed about the status quo of the tissues they reside in.
They're like little security guards, always checking to make sure everything is in order.
Exactly.
And where are these SCINT1 and BTNL molecules found?
Well, SCINT1 is expressed in the skin,
Yeah.
and DTNLs are found in a variety of tissues, including the gut.
And they play a role in regulating gamma delta T cell residency and development in those tissues.
Yes. They provide homing signals, guiding the gamma delta T cells to the right place. And they help to maintain the right balance of gamma delta T cell populations in those tissues.
That makes sense. So what happens if the expression of these ligands is disrupted?
Well, that can lead to problems. For example, in patients with celiac disease, the expression of BTNL molecules in the gut is disrupted, and that leads to a shift in the gamma delta T cell populations, which can contribute to the disease.
That's interesting. So those ligands are really important for keeping things in check.
They are, and it's not just SCINT1 and BTNLs. Gamma delta T cells also express a variety of coreceptors on their surface, and these coreceptors can either amplify or dampen the signals that the gamma delta TCR
So
receives.
they're kind of like volume controls.
Exactly. And these coreceptors allow gamma delta T cells to fine-tune their responses to different threats.
Can you give us some examples of these coreceptors?
Sure. One example is NKG2D. It's a coreceptor that's expressed on gamma delta T cells in the gut and skin, and it binds to ligands that are expressed on stressed or damaged cells. When NKG2D is activated, it triggers the gamma delta T cell to release cytokines that help to protect the tissue.
So it's like a distress signal.
Exactly.
What are some other examples?
Another example is the CD94 NKG2 complex. This complex can either activate or inhibit gamma delta T cells, depending on which NKT2 molecule is paired with CD94. During inflammation, the activating form of this complex is often upregulated on gamma delta T cells, which enhances their effector function.
So it's like a switch that can flip them from a resting state to an activated state.
Exactly.
Are there any other coreceptors that are important?
There are many, but one more worth mentioning is semaphorin 4D, or CD100. It's a costimulatory molecule that interacts with plexins on epithelial cells and can actually promote wound repair by gamma delta T cells.
Oh, wow. So they can actually help to heal tissues.
They can. And then there's Li49E, which is an inhibitory coreceptor that's mainly found on mouse gamma delta T cells. It helps to keep them in the tissues where they're needed.
So these coreceptors really play a critical role in shaping the gamma delta T cell response.
They do. They add another layer of complexity to an already complex system.
Now, one of the things that gamma delta T cells are known for is their ability to produce IL-17.
That's right. IL-17 is a cytokine that's heavily involved in inflammation, and gamma delta T cells are one of the major sources of IL-17 in the body.
And what's interesting is that they don't necessarily need the same signals that alpha-beta T cells need to start producing IL-17.
Exactly. Their IL-17 production is regulated by a transcription factor called ROR gamma T. And ROR gamma T is expressed in gamma delta T cells very early on, even before they leave the thymus.
So they're kind of primed to produce IL-17 right from the start.
You could say that. And in mice, IL-23 can actually amplify IL-17 production by gamma delta T cells.
IL-23, that's another cytokine, right?
Right. It's a cytokine that's involved in promoting inflammation.
And what about in humans? Is IL-23 important for human gamma delta T cells?
It is, but it's not always required for the initial secretion of IL-17. IL-23 can, however, help to maintain the population of IL-17 producing gamma delta T cells.
So there might be other earlier triggers that we don't fully understand yet.
That's a possibility.
Now, what are the consequences of all this IL-17 production?
Well, in the skin, IL-17 can drive keratinocyte proliferation.
Keratinocytes, those are the cells that make up the outer layer of our skin, right?
Yes. And when keratinocytes proliferate too much, it can lead to the formation of psoriatic plaques.
So IL-17 from gamma delta T cells could be a key driver of psoriasis.
It's definitely a possibility.
And it's not just the skin. The article also mentions that gamma delta T cells can circulate between tissues in inflammatory environments.
Right. So if they're activated in one tissue, like the gut, they can travel to other tissues, like the skin or joints, and spread the inflammation.
It's interesting. So they're like little inflammatory messengers.
In a way, yes.
The article goes on to talk about the IMQ mouse model. Can you explain what that is?
Sure. The IMQ mouse model is a way to study psoriasis in mice. Basically, they apply a cream containing a compound called imiquimod to the skin of the mice. And imiquimod activates certain immune receptors on the skin, which leads to inflammation and the development of psoriasis-like lesions.
So it mimics what happens in human psoriasis.
Yes. And in this model, gamma delta T cells have been identified as the major source of IL-17.
So they're really driving the inflammation in this model.
They are. And what's even more interesting is that when researchers deplete gamma delta T cells in these mice, the severity of the psoriasis-like disease decreases significantly.
That's pretty strong evidence for their role in psoriasis.
It is, and there are some therapeutic approaches that are being developed to target gamma delta T cells in psoriasis.
Oh, really? Like what?
Well, one approach is to inhibit ROR gamma T.
Yeah.
As we talked about earlier, ROR gamma T is the transcription factor that regulates IL-17 production by gamma delta T cells. So if you can block ROR gamma T, you can reduce IL-17 production and potentially ameliorate the disease.
That makes sense.
Another approach is to inhibit Bruton's tyrosine kinase, or BTK. BTK is an enzyme that's involved in signaling pathways downstream of the gamma delta TCR. So by inhibiting BTK, you can block the activation of gamma delta T cells and reduce their production of IL-17.
So there are a couple of different ways to target these cells.
Yes. And there are also some regulatory molecules that can help to keep gamma delta T cells in check.
Oh, really? Like what?
Well, one example is CD109. It's a protein that's expressed on keratinocytes, and it can actually inhibit the activation of gamma delta T cells.
So it's like a brake pedal for these cells.
Exactly. And there are other regulatory molecules as well, like CD30 and its ligand CD30L, as well as the cytokine IL-38.
So there's a whole network of checks and balances that help to control gamma delta T cells.
Yes. And when this network is disrupted, it can lead to problems like psoriasis.
Now, what about in humans? What have studies shown about the role of gamma delta T cells in psoriasis?
Well, in humans with psoriasis, researchers have found that V gamma 9 V delta 2 T cells accumulate in the psoriatic lesions. And these cells produce a whole bunch of pro-inflammatory cytokines like IL-17A, IFN gamma, and TNF-alpha.
So they're really contributing to the inflammation in those lesions.
They are, and interestingly, these same cells are often decreased in the peripheral blood of psoriasis patients.
So it seems like they're being recruited from the blood to the skin.
Exactly. And there's another subset of gamma delta T cells called V delta 1 negative, delta 2 negative, gamma delta TCR high T cells that are enriched in patients with more severe psoriasis.
So the more severe the disease, the more of these cells you see.
Right. And researchers have also found that a molecule called butyrophilin 3A1 or BTN3A1 is overexpressed in psoriatic lesions.
We talked about BTN3A1 earlier.
Yes, we talked about it in the context of the gut. But it turns out that BTN3A1 is also expressed in the skin. And when it's overexpressed, it can actually promote the activation and proliferation of V gamma 9 V delta 2 T cells.
So it's like adding fuel to the fire.
Exactly.
Okay, so we've talked a lot about the role of gamma delta T cells in psoriasis. Now let's shift gears and talk about spondyloarthritis.
Okay. Spondyloarthritis, or SPA, is a group of inflammatory diseases that affect the joints. And it includes conditions like ankylosing spondylitis, psoriatic arthritis, and enteropathic arthritis.
And what role do gamma delta T cells play in SPA?
Well, in ankylosing spondylitis, researchers have found an increase in the number of IL-23 receptor-positive gamma delta T cells in the blood.
So these cells are responsive to IL-23.
Right. And they produce IL-17 in response to IL-23.
So they could be contributing to the inflammation of the joint.
Exactly. And increased levels of IL-17 producing gamma delta T cells have also been found in the blood and synovial fluid of patients with other types of SPA.
Synovial fluid, that's the fluid that lubricates the joints, right? So these cells are definitely present in the inflamed joints.
They are. And there's growing evidence to suggest that they're playing a role in driving the inflammation.
Now, the article talks about the gut-skin-joint axis. Can you explain what that is?
Sure. The gut-skin-joint axis is the idea that inflammation in one of these areas can influence the other two.
So, for example, if you have inflammation in your gut, it could trigger inflammation in your joints or skin.
Exactly. And there's a lot of evidence to support this idea. For example, inflammatory bowel disease, or IBD, is more common in people with ankylosing spondylitis. And conversely, people with IBD are more likely to develop joint problems.
So there's definitely a link between gut and joint inflammation.
Yes. And there's also a link between skin and joint inflammation. Many people with psoriatic arthritis also have psoriasis.
So is there any evidence that gamma delta T cells might be involved in these connections?
Well, one possibility is that gamma delta T cells are activated in the gut by exposure to certain microbes or other inflammatory stimuli. And then these activated gamma delta T cells can travel through the bloodstream to other tissues like the joints or skin, where they can promote inflammation.
So they're like little seeds of inflammation that can spread throughout the body.
That's a good analogy.
And what about the enthesis? What is that?
The enthesis is the point where a tendon or ligament attaches to a bone,
Okay.
and it's a common site of inflammation in CPA.
So could gamma delta T cells be involved in enthesitis?
Yes, it's possible. The enthesis is a site where there's a lot of mechanical stress, and that stress can trigger inflammation. And as we talked about earlier, gamma delta T cells are very good at responding to danger signals, including mechanical stress. So it's possible that they're being activated in the enthesis and contributing to the inflammation.
The article mentions a study that looked at the role of gamma delta T cells in enthesitis in mice.
Right. They found that a specific population of gamma delta T cells that express the IL-23 receptor were present in the enthesis. And these cells were producing IL-17 and IL-22, which are both pro-inflammatory cytokines.
So these cells could be driving the inflammation in the enthesis.
They could be. And what's interesting is that these cells seem to be independent of conventional Th17 cells.
Th17 cells, those are another type of immune cell that produces IL-17, right?
Right.
So this suggests that gamma delta T cells might be playing a unique role in enthesitis.
That's right.
And what about in humans? Have they found similar cells in the human enthesis?
They have. They found a population of V delta 2 T cells in the human enthesis that express ROR, gamma T, and CCR6, which are both markers of Th17 cells.
So they have a similar profile to the cells that were found in the mouse enthesis.
They do. And these cells also produce IL-17 and IL-22.
So they could be playing a similar role in human enthesitis.
They could be.
Now, the article also talks about the synovium.
Right. The synovium is the lining of the joint, and it's another site of inflammation in SPA.
So are gamma delta T cells found in the synovium?
They are, and in the synovium of ankylosing spondylitis patients, they found a population of gamma delta T cells that co-express IL-17 and GM-CSF.
GM-CSF, what's that?
GM-CSF is another pro-inflammatory cytokine, and it's been linked to the erosive phase of SPA.
So these cells could be contributing to the damage that's seen in the joints.
They could be. And they also found an increase in the number of IL-23 receptor positive V delta, 1 positive IL-17 producing T cells in the blood of patients with axial SPA.
Axial SPA, that's the type of SPA that affects the spine, right?
Right.
So there are different subsets of gamma delta T cells that seem to be involved in different aspects of SPA.
Yes, and it's important to note that gamma delta T cells don't act in isolation. They interact with other cells in the joint, including stromal cells.
Stromal cells, those are the cells that make up the connective tissues in the body, right?
Right, like fibroblasts and osteoblasts. And gamma delta T cells can activate these stromal cells and make them produce pro-inflammatory cytokines.
So it's really complex interplay of different cell types.
It is, and it's still not fully understood.
Now, the article mentions a couple of potential therapeutic targets for SPA that involve gamma delta T cells.
Right. One target is ROR gamma T. As we discussed, ROR gamma T is the transcription factor that regulates IL-17 production by gamma delta T cells. And there are drugs that are being developed that can inhibit ROR gamma T, and these drugs have shown some promise in reducing IL-17 production and inflammation in animal models of CA.
So they could potentially be used to treat CPT in humans?
They could be, but more research is needed.
And the other target is JK kinases.
Yes. J kinases are enzymes that are involved in signaling pathways downstream of many cytokine receptors, including the IL-23 receptor. And there are drugs called JAK inhibitors that can block the activity of these kinases.
And JK inhibitors are already being used to treat some inflammatory diseases, right?
They are. They're being used to treat rheumatoid arthritis and psoriatic arthritis.
So they could potentially be used to treat SPA as well.
They could be. And there are clinical trials underway to test the efficacy of JK inhibitors in SPA.
So there's hope for new treatments for SPA on the horizon.
There is.
Now, before we wrap up, I wanted to touch briefly on the role of gamma delta T cells in the central nervous system, or CNS, and the eye.
Okay.
The CNS is traditionally thought of as being immune privileged.
Right, meaning that it's protected from the immune system.
Well, exactly. But it turns out that gamma delta T cells are present in the CNS.
They are, and they seem to play a role in both health and disease.
How so?
Well, in healthy individuals, gamma delta T cells in the CNS have been shown to contribute to neuroplasticity and memory formation, and they do this through the production of IL-17.
Wow. So they're actually involved in brain function.
They are, but in diseases like multiple sclerosis or MS, gamma delta T cells seem to play a more detrimental role.
How so?
Well, in MS, there's an autoimmune attack against the myelin sheath that surrounds nerve fibers in the CNS. And gamma delta T cells have been found to accumulate in the lesions that form in the CNS in MS. And they've also been shown to produce pro-inflammatory cytokines that can damage the myelin sheath.
So they could be contributing to the progression of the disease.
They could be. And there's also evidence to suggest that they might be involved in the development of other autoimmune diseases, like type 1 diabetes.
Wow. So they're really versatile cells.
They are.
What about the eye? What role do they play in the eye?
Well, the eye is another immune privileged site, but gamma delta T cells are present in the eye as well.
And what do they do there?
Well, in healthy individuals, they seem to play a role in maintaining immune tolerance in the eye. Specifically, they're involved in a process called anterior chamber associated immune deviation, or ACADE. And ACADE is a mechanism that prevents the immune system from attacking antigens that are present in the anterior chamber of the eye.
So they help to protect the eye from inflammation.
Exactly. But in diseases like uveitis, gamma delta T cells can actually contribute to the inflammation.
Uveitis, that's inflammation of the uvea, which is the middle layer of the eye, right?
Right. And uveitis can be caused by a variety of things, including infections and autoimmune diseases.
And how are gamma delta T cells involved?
Well, they've been found to accumulate in the uvea, in the uveitis, and they produce pro-inflammatory cytokines that can damage the eye.
So they can be both good and bad for the eye, depending on the context.
Exactly.
Well, this has been a truly fascinating deep dive into the world of gamma delta T cells. It's amazing how much we've learned about these cells in recent years, but it's also clear that there's still a lot that we don't know.
That's right. There's still a lot of research that needs to be done to fully understand the role of these cells in health and disease.
But one thing's for sure, these cells are incredibly versatile. They can play a role in just about every organ system in the body, and they can be both protective and pathogenic, depending on the context.
Exactly. And that makes them really exciting to study.
So to wrap up, I just wanted to reiterate some of the key takeaways from today's deep dive. Gamma delta T cells are a unique subset of lymphocytes that have both innate and adaptive immune functions. They're found in a variety of tissues throughout the body, including the gut, skin, joints, CNS, and eye. And they play a role in both maintaining health and driving inflammation. Their activation is often independent of traditional MHC presentation. And they can produce a variety of cytokines, including IL-17, which is a key driver of inflammation in many diseases. There's growing evidence to suggest that gamma delta T cells are involved in the gut-skin joint axis, and they may be a key link between inflammation in these different parts of the body. There are a number of potential therapeutic targets for diseases that involve gamma delta T cells, including ROR gamma T and JK kinases. And finally, there's still a lot that we don't know about gamma delta T cells, but research is ongoing, and new discoveries are being made all the time.
It really makes you wonder, doesn't it?
What's that?
Well, given the diverse and sometimes opposing roles of gamma delta T cells, how might future therapies selectively modulate their beneficial functions while suppressing their detrimental contributions in diseases like spondyloarthritis and psoriasis? It's a challenge for sure, but it's also an incredibly exciting opportunity.
Absolutely.
So for you, our listeners, what do you think? Leave us your thoughts in the comments below.
And don't forget to subscribe to the Deep Dive for more deep dives into the world of science and medicine. We'll see you next time.
Bye!